Good morning. My name's Owen Jones. I'm a PhD student at the Great Ormond Street Institute of Child Health, which is part of University College London. And today I'm going to talk to you about my PhD project, which has been towards improving the integration of stem cell-derived photoreceptor transplants into the retina. As I'm sure you're all aware, the part of our eye that detects light is called the retina. And as light enters the front of the eye, it gets focused onto this layer which sits at the back of the eye. Within the retina are these specialized nerve cells called photoreceptors, which act as sensors to detect the light. And they're arranged in a layer, which is the first of 3 layers of different nerve cells that make up the retina. We have 2 flavors of photoreceptors, one type called the cone photoreceptors and a second type called the rod photoreceptors. And they're shown in red and green in this diagram. They're crucial for our vision. After detecting light, they transmit information about it, such as its brightness, to the second layer of nerve cells. And these cells then relay the information to the third layer, the retinal ganglion cells, to the brain, which allows us to build an image of the world that we perceive around us. You might wonder how these cells pass information to each other. Well, the layers of nerve cells are actually connected to each other with these long projections. And they can be thought of like the cables between pylons carrying electricity to a town, the town being the brain where the information ends up. In diseases like macular degeneration, these photoreceptor cells unfortunately die. And this means that they can no longer detect the light or pass information into this circuit. And there are several other diseases where the rod photoreceptors, or both the rod and cone photoreceptors, die. Unfortunately, this is irreversible, but the second and third layers of nerve cells in the circuit survive. So an aim within Professor Souden's lab and in other research labs around the world is to develop a therapy whereby we can replace these photoreceptors with healthy ones that we've generated from stem cells in the lab. If we're able to do this and that they're able to detect light and crucially connect with the next nerve cells in the circuit, then we might be able to rescue the function. So first I'm going to tell you a little bit about how we grow the retina from stem cells in the lab. A stem cell is a cell type that can become any other type of cell, and they're very useful in lots of areas of research, such as liver, heart, and gut research. But in Professor Souden's lab, we've developed a method of turning these human stem cells into retinal cells. So to do this, what we do is we grow the stem cells, which are human stem cells, first in a little petri dish, and then we place them into this tiny little well. You can imagine the well as like a cup. And we feed the cells within the cup with the ingredients that they need to become retinal cells. And this is a process that takes around half a year because the cells develop in the same rate as they do in the baby's eye before birth. Feeding the stem cells to become retinal cells requires a lot of effort and takes several hours on a daily basis. But provided that we get it right along that long process, then we get these little balls of cells that are made up of all of the cells that are in the retina. They look like mini eyes, but it's more accurate for us to describe them as mini retinas because they don't have the other cell types that make up the eye. Among these nerve cells in the retina are the photoreceptors. So our mini retinas also contain these 2 flavors of photoreceptors, the rods and the cones. These are actually really similar to the human photoreceptors in the healthy eye. So this gives us an opportunity to use them for research, but also potentially in the future, therapeutically. Because they're so similar, we hope to be able to extract them from the mini retinas and then transplant them into the patient's eye. An ophthalmologist can already perform vitreoretinal surgery to introduce new cells into the eye with relative ease, but the hold-up is that the efficacy of this therapy is currently limited. So this means how well the cells are able to recover the light sensitivity of the retina. A reason we think this might be is that the transplanted cells struggle to connect with the second nerve cell in the circuit. So scientists in the lab are working on strategies that will encourage the connection of the photoreceptors with the second nerve cells. And this is the issue that I've been focusing on in my PhD project in the last 4 years. To understand how we might encourage these photoreceptors to connect, I first researched what we know about how they form with their connections in the retina and the developing eye before birth. And previous experiments have shown us that these cables that are sent out from the photoreceptors towards the second layer of nerve cells get sent out during development in this sequence where they reach out to the second cell, and in turn, the second cell reaches out to them. When they meet in the middle, they connect, and that connection is what's really important for them to form that circuit that transmits information to the brain. So the start point of my study was wondering whether we can encourage those projections, those cables that allow for the connections to be built. If we can encourage the growth of those projections, then we might be able to encourage more connections in the transplant. So I set out with the aim of identifying if I can use some drugs that may cause photoreceptors projections to grow. And they've been shown to have that effect on other nerve cells in the past. Not retinal nerve cells, but other nerve cells in the lab. And these drugs are already clinically approved, so it's theoretically possible for us to use the drugs along with the stem cells in order to improve the success of the transplant. So my objective was to investigate whether photoreceptors in the lab grown in a dish grow these projections, and then to test whether some drugs that we thought might be able to encourage the projections to grow did have that effect. So how I did this was I first grew my mini retinas for 20 to 30 weeks, which all of my friends think is a bit crazy that I sit in the lab and grow my mini retinas for this long. But then I extracted the cells from them, which involves breaking them apart to separate the nerve cells. And then I transferred them to one of these dishes. And the dishes have hundreds of these little wells that I was describing earlier. But instead of having the stem cells in these wells, this time I had the retinal cells. I did this several times until I was confident that I could look at them down the microscope and study what was going on with their projections. And then when I was confident in my method, I treated them with hundreds of different drugs at different dosages, taking advantage of the hundreds of wells that we have in this dish that has hundreds of little wells. So doing that, I was able to then preserve the cells. I have to fix them, preserve them, and take them to stain them to look for specifically the photoreceptors. So I stain the photoreceptors with something which specifically labels them. And that allows me to look down the microscope at only the photoreceptors to understand and look at their projections. So I took hundreds of pictures of them treated with the hundreds of different combinations of drugs and dosages. And then I tried to analyze these images to identify whether some of the projections were longer than others with different treatments. If I was to do this manually by looking at all the images, it would take me years and years. So I had to use a clever method which uses the computer and an algorithm built in the computer to detect the photoreceptors and measure their projections. So what I'm showing on this slide is in blue all of the different cells that are within the mini retinas that have gone into my dish. And in purple, I'm showing you the stained photoreceptors. And you can see that they've got these long projections coming from them. So the computer algorithm that I described enables me to identify thousands of these cells across hundreds or thousands of images, and then to measure how long each of their projections is, and then to get that information out as numerical data that I can analyze and look for differences between. So by analyzing these experiments, what we found was that surprisingly, the photoreceptors not treated with drugs were able to grow these projections. They have the same appearance as photoreceptors do sometimes in the human retina. And this is good because it indicates that after breaking them apart from the mini retina, they're still capable of sending out their projections and hopefully forming the circuit that they need to in order to be used as a cell therapy. The experiment also allowed me to identify 2 drugs that encourage the growth of longer projections from these cells. On this slide, I'm showing that this long projection from the purple photoreceptor cell body, which is down at the bottom in the left, and you can see this long process that's grown out, the long projection that's grown out from the cell, presumably looking to connect with other cells in the dish. And the same was true of a second drug called blebistatin. So the first drug is called the ROCK inhibitor, or Rho kinase inhibitor, and the second drug, blebistatin. Blebistatin caused these long and winding projections, looking as though they are finding other cells within the dish, which is very interesting and a good finding. So here I'm showing a graph of the numerical data that I've analyzed from that experiment. And the white bar is showing the length of the control treated photoreceptors. So these were long, but they weren't as long as after treatment with the 2 drugs, which are the blue and the yellow bars. So you can see that there was an increase in the length of the photoreceptors. And this is the effect that we were hoping for. So the next thing I'm going to talk about is a clever technology that we've used to further investigate how the photoreceptors are forming those projections. What we've done is that we've put some genes from a jellyfish which make live rods glow green and live cones glow red within our mini retinas. And this means that instead of preserving the cells and staining them as I described that we have to do, we can actually look at them in real time and study how their projections are growing in real time. This is really exciting because it hasn't been done before. When we grow the mini retinas with these genes from the jellyfish in them, we can look at the fluorescent rod photoreceptors growing at the surface of the mini retina and the fluorescent cone photoreceptors growing at the surface of the mini retinas and study how they're changing over time. And if we look with a very powerful microscope, what we see is that the shape of the photoreceptors at the surface of the mini retina is actually really similar to what we know the photoreceptors shape to be in the human. And this gives us confidence that the cells are very similar to the photoreceptor cells in the human retina. So on this side, you can see the long, we call them segments that come from the photoreceptors. And these are actually the part of the photoreceptors that allow for light to be detected. This is really cool and gives us confidence that the cells are going to be similar to the human photoreceptors, giving us confidence that they'll be able to be used in a transplant. So I took these mini retinas, which have the glowing rods and cones in them, and I did the same experiment where I broke them apart to extract all of the different nerve cells. But instead, this time I have my glowing rods and glowing cones. I was able to transfer those into a similar multi-weld dish and look at them down the microscope. This microscope was more advanced, and it allows me to take a time-lapse series of images so that I can construct a film, if you like, of the cells making their projections over time to understand what's going on better. And what these experiments showed is actually really fascinating, because the nerve cells, including the fluorescent cones and rods that are here moving around in the dish, these are the first movies that we've captured of cells, of the photoreceptor cells in a dish doing this. What you'll appreciate is that along with the photoreceptors, there are lots of other types of cells. So these are the other pylons, if you like, that are important for forming connections, forming circuits. Oh, my movie's not working. Forming connections in the retina. And these other cells perhaps are what the photoreceptors are moving around to find in the dish. So here's another movie of that happening, where the green rods and the red cones are moving around in the dish, and the blue other cells, the other pylons, are moving towards them in order to find them and interact with them and connect with them. So I was very interested in trying to understand a bit more about what's going on here. Is it that the cone and rods are reaching out with their projections to the other blue cells, or is it that the other blue cells are reaching out more to them? By studying lots and lots of these time-lapse images, my findings are that the rod and cone photoreceptors mostly require contact with the other cells in the dish in order to form their projections, indicating that those other cells are really important for the circuit to be built. So here I'm showing a time-lapse series at the 1st hour, the 5th hour, the 9th and the 13th hour. And what it shows is that the photoreceptors are growing processes, growing these projections, but they are sitting right next to other cells. So these small dots next to the photoreceptors are other cells. And this was most often the case. So when they were sat next to these other cells, they were better able to send out their projections. But very interestingly, the cone photoreceptors, which now I'm showing at the bottom of the screen, the cones were able to grow their projections without contacting any other cells, seeming to show that they were looking for or seeking out the second nerve cells to form the connections with them. So at the 5th hour, you can see this long process from the cone. This is really encouraging because the cones are obviously important in the function of the macula. They're the cells that allow us to detect light and give us our central vision. So knowing that they're able to send their projections out towards the second nerve cells in the circuit is very promising, as it indicates that after being transplanted, they'll be able to do this. And perhaps our small molecules will be able to, our drugs will be able to encourage how well they do this. So to sum up, what my PhD project has shown is that the photoreceptors grow projections in the dish. And by doing that big experiment with hundreds and thousands of images, I was able to show that 2 drugs were able to enhance the growth of the photoreceptors projections. And these were blevastatin and the rho kinase inhibitor. The time-lapse images showed us that the other cells in the mini retinas help photoreceptors to grow their projections. But very interestingly, that the cone photoreceptors can grow projections on their own. So in the future, we're looking to test if the drugs can improve the efficacy of the transplants in animals. So we've shown that they can increase the projections length in the dish, but now we're going to try and understand if they can improve the efficacy of the photoreceptor transplant. We hope that they will be able to do this by encouraging the length of the projection to grow, and hopefully that will mean that more connections form between the photoreceptor and the second nerve cell. The second thing we're trying to do is to understand how the other cells help the photoreceptors grow projections in those time-lapse movies. This might give us clues as to why the other cells are important and how we can use features of those other cells in order to further encourage the connection between the transplanted cells and the second nerve cells in the recipient of the transplant. And that just leaves me to sum up and thank all of the members of the Soudan group who've helped with this work, and all of the staff at Great Ormond Street Hospital who have contributed to helping me learn and develop in the last 4 years of my project. It's been very enjoyable. And particular thanks to the Macular Society for funding my research and providing support throughout. And thank you to all of you for listening. I look forward to answering your questions during the Q&A afterwards. Thank you.